I was asked just to first quickly start off with a bit about what I'm doing now and where I'm at now, and then I'll kind of get into more about what I did for my master's work down in Calgary. So, as was mentioned, I'm Tara, I'm from Ottawa, did my master's in Calgary, and now I'm actually based in New Zealand, in Auckland. And I'm doing stuff kind of along similar lines as my master's work, and that's kind of what got me here. But it's actually working with scallops down here, and I'm seeing if we can spawn them and get them to grow in their early life stages so that we can use them for different kinds of restoration efforts and aquaculture purposes and stuff like that. So I'm right in the thick of my experiments right now, and we're kind of in the middle of the day, so I have some scallops in the lab running through experiments as we speak. But yeah, it's really great, and all of my work on my master's is definitely what got me here, and so I'm excited to kind of fill you all in on what we did there. So getting kind of right into it, my master's was about using these in-stream incubation capsules and figuring out if we could use that type of method to do something called a conservation translocation in Alberta. So I'll get into more details about what that all means in a second. But starting off, this work would not have been possible without the help of so many different people and organizations, and the ones listed here alone are definitely, it's not all inclusive, but this is just an idea of how many people it took to pull this off, and it was a very big feat, but we're all very proud of the work now. So I'll run through some quick background on bull trout, just the stuff that's relevant and important that you know, project relevant stuff, but I'm sure you all know and love the Alberta bull trout. But for my master's project specifically, we worked with the Alberta bull trout, but they are native to northwest of North America, so they run into the States as well. Sorry, I'm just making sure that this is all the same. There we go. They do have some specific habitat requirements, which make them very helpful in determining ecosystem health, but it also makes their habitats quite limited. So they require things like cold, connected, and complex habitats. And generally, we were working with the abfluvial bull trout, so they lived in lakes and then they swam up streams to spawn. And the ones that we caught and handled specifically for this work were on average 657 millimeters long, and they weighed on average 2,766 grams. So they were fair-sized bull trout that we were handling, which was great for the research and what we were using them for. As for their status right now, Canada makes up about 80% of their total range. As you can see on this map, they obviously don't take up it's not consistent throughout the range because streams and bodies of water are not everywhere throughout the range, but that is the general idea of their range. And we were working specifically with the Saskatchewan Nelson population, which contains that adfluvial population that I was talking about just earlier. And based on the most recent population estimates of the creek, that specific population was considered to be stable, although in Alberta they're considered threatened, and they were listed in 2019 on the Species at Risk Act across the country. So in general, we were working with a stable individual population, but the population as a whole is not doing great. And some of the things that are posing big threats to them are climate change, habitat fragmentation, hybridization with things like the brook trout, and competition with those species as well. And some things that we're doing to try and help them include those angling restrictions and stocking projects and removal of some non-native species. As for their reproduction, which is more specific as to what we worked on, they migrate to headwaters for spawning. So for my project, that was Smith Dorian Creek in Kananaskis. And they tend to spawn mid-August to late October, and then they incubate in stream over winter and then swim up. So the little tiny baby fish swim up in early spring, and they tend to reach maturity and start spawning at around 7 years old. And females tend to produce about 2,000 eggs each. So then I'll just get right into my main focus of my work. So the kind of main idea was to assess whether or not we could use these in-stream incubation capsules for conservation translocations. And then with that, and in doing that, we kind of wanted to look at different things like temperature and sediment and freezing to see if those would potentially impact how successful the in-stream incubation capsules were. And so I'm just going to run through these quickly because it's just kind of summarizing them. So we looked at whether the capsules were successful or not, and if we had alvin survival. And then we also looked at whether there were groundwater upwellings, freezing events, or fine sediment impacts on the survival of the alvin. So I'll get into kind of the results later, but those are the 4 main things that we looked at in my research. A timeline of what we did, just to give you kind of an idea of where we were at a couple years ago. So we started in 2023 in kind of the spring and summer, and that was when we started scoping out the sites, installing temperature loggers, getting everything ready to go. And then during the spawning event in fall 2023 is when we caught the fish, collected the eggs, did the fertilization, and deployed all the capsules. So that was kind of the major field work starting off the experiments. And then over the winter, I did some monitoring of the sites, some write-up, and then into early spring, we collected all of the data, and then I kind of wrapped it up and did some data analysis to figure out and give you guys the results that we found. So we ended up choosing Smith-Dorrien Creek as our site, which is just kind of outside of Calgary area. So you can see Calgary on the right there. And then that cluster of dots is where our site was located. It's quite a critical site in kind of the bull trout world in Alberta. It's an at-risk population range, but the population itself quite stable, as I mentioned. And it's also quite a frequently studied site and population of bull trout. So we were able to have some background information and history on the population to kind of build off and know rather than just going in blind. So we had important information like historical red data available to us that we could use to base our site and site selection on. And there was also an egg take, so similar to what I'll get into in a few slides, done there in 2021 by the Department of Fisheries and Oceans. So we had some hands-on experience of people that we actually knew and could talk to get that knowledge and figure that out. And then specifically at the Smith-Dorrien Creek site, I used that historical red data to help select where we were going to put the tubes in the stream. So I'll again get into it a bit farther on, but the 2 red sites, so 1 and 4, we ended up not using because of some typical fieldwork struggles. So we ended up using and burying eggs where the number 2, 3, and then the trench is located. And it was more or less supposed to represent a distribution kind of based on historical red densities. So where there was once lots of reds in those red surveys or less reds to kind of see the distribution. But we ended up getting the upper and mid range of red densities, which was helpful to tell us if the tubes were really going to work or not, because we knew bull trout have spawned there in the past. And then getting into these capsules that I've kind of been mysteriously talking about, we built them ourselves using prefabricated mesh hatchery tubes, which I usually use to transport eggs. But we cut them down to size, and we kind of determined that size to maximize space while also limiting the kind of risk of shearing from ice and movement and everything like that. So they got down to about a hockey puck size, and that's what we ended up calling them throughout the project. And we filled them with aquarium-sized gravel that we just rinsed through to give the eggs some more security so they weren't being jostled around in those tubes. And then we made those caps. So those caps were solid to begin with, and we drilled those holes by hand to, again, allow for water flow and nutrient flow all throughout. So those eggs were not just trapped in there, they had access to the stream bed beyond. And so getting into our field work and that big time that I talked about in the fall of 2023. Yes, 2023. So we have electrofished 3 sections of the creek, which is what they had done in 2021. So we kind of had that guidance to lead us. And so we started at James Walker Creek and went above and below where that creek met. And we ended up catching more fish than we needed. So any fish deemed spent or not mature, they were immediately released. And we ended up keeping a total of 8 males and 8 females for this research, as well as some research that we kind of split the eggs off with DFO to run a separate project on temperature stuff. And the fish that we caught were held in stream overnight in bags. And then we came back out and got into it the next day, where we used an ultrasound to be able to confirm that there were eggs still in the females. And all females and males, they were expressed of their gametes. So we did that by lightly pressing on their abdomen and just caught them, which is quite cool to watch. There are some videos out there I recommend. And so we crossed the eggs of the females and the males, and they were all mixed together to water harden. And then we randomly counted them out individually into these tubes that I showed you. And we put 50 eggs in each tube, and then we got right into capsule installation because we were quite limited with day. And once those eggs were fertilized, we had to get them out into the field that day to kind of minimize any risk that we put onto them. So then we get into capsule installation. So that was at those 2 sites that I told you about, site 2 and 3. And they each got 36 capsules each, clustered in 6s throughout that section of stream. And we buried them at varying depths to see if depth had an impact on survival. And we did this by kind of an interesting tactic that you can see in the picture on the right. So we had a PVC pipe that once we dug to a certain depth, we shoved the PVC pipe in to hold that depth so the hole didn't backfill. And then we dumped water down the tube, dropped the tube down the PVC pipe, and then covered it back up with the sediment. It was a very lengthy process to dig 36 holes at each site. And it was, with the water flowing over the holes and everything, it took a lot longer than we were expecting. So that kind of led us to having to install a trench. So initially we had planned on burying 4 sites, and it got to the point where we were running out of daylight, and we had 72 capsules that we had to get in the water because they wouldn't have been effective and survived the next day. It would have impacted the alvin survival. So we knew that we had to get those in the water, and we came up with this kind of last minute idea. I remember me and Jess standing in the creek talking about this. And then, so we came up with this trench. And as you can see on the picture on the right, again, we dug the trench to all the same depth in this picture to start with, and just started layering the capsules in there all in the same place. And it actually ended up providing an interesting comparison between methods. So we had those individually placed capsules, but then we also had a more kind of human effective management perspective, useful method of digging all at once in a trench. So we ended up going back and moving depths. So we had 2 depth representatives, similar to the individual sites. And the location of the trench was in between the 2 where we had buried the capsules individually. So it was more or less representative of where reds have historically been. And so then we get into, once we wrapped that all up, all the eggs were in the stream. We, as you can obviously know, we couldn't go out and check on them every single day and open the eggs because then we would be disturbing them. So we also had things like the hatchery where we're growing them, and we also did these monitoring visits. So we removed some capsules monthly about from the trench because they were all in one cluster to look at developmental analysis. So I was able to look at pictures and kind of estimate timing to pull them out. And we also had temperature measurements in the water at surface at 20 centimeters and at 30 centimeters, so at all the depths where those capsules were buried. And then I also had an undergrad who helped me out and did some substrate composition. So she was looking at kind of the creek bed and what the makeup of the substrate was in the areas that we were putting the tubes to see if that also had an influence on the survival. And then based on those periodic visits to the trench, we were able to decide when we should go out and collect our tubes. So all capsules were collected in March 2024. And it was again a big effort from everybody who was willing to come out and help us in lots of snow. And it was quite an effort to find the eggs that were buried individually because we had not much of a great way of identifying and finding those sites again. So we were just digging up the entire creek bed. And then we opened each capsule up individually and counted any dead or unhatched eggs to determine how many had survived. We also counted how many alvin were in there. So you can see in this picture that we have alvin in the picture and we also have eggs in the picture. That's a picture from February. So we weren't quite at full development yet, which is why you can still see some eggs with some fish in it. But once we got to full development, they were either dead eggs, obviously dead eggs. There was none that were partially developed. And any fish that were still in the capsule, so as alvin, once they reached full development and we were taking them out, they were released back in the stream. And we also went out there and measured groundwater at each individual location where there was a tube buried, and we collected all of our temperature loggers and data. And here I just have kind of a quick run through of where all the capsules went, just to give you a better understanding, because sometimes explaining it by words just doesn't quite hit the mark. So we sent 3 off to the lab to run some fertilization tests. And then we had a total of 144 go into Smith Dorian Creek. So we had 36 at site 3 and then 36 at site 2 in individual sites. And then the trench was very close to site 2, so we grouped it together there. And that's where the remaining 72 went. And then, as you can see, we pulled them up a few from the trench for those monitoring visits. And then you can see in the end that we actually tended to struggle with some recovery, particularly in the individual site deployment. So here, it kind of helps you visualize the retrieval of the capsules. So we had 100% recovery in the trench. We went and we found all the tubes. They were all right where we left them. Versus 68% recovery in the individual sites. So this is specifically site 3 and site 2 on the screen here, and any of the grayed out ones we weren't able to find again. And then the green ones had above 90% survival, and then the red ones had below 50% survival, but there was still survival. And we had about 2 to 6 tubes recovered per clustered group. And I'll get into our survival now. We had, so site 2 we found had highest survival, which kind of gave us some great insights into future use of these tubes and in-stream incubation, because it gave us some solid results to build off of. So I'm just going to focus here on the highlighted red range. That's kind of all that you need to care about. The rest is in count values, which won't really make sense easily because the count is out of 50, but the red is in percentages. So as you can see, site 2 had the highest maximum, but also the lowest minimum. And I'll get into that a bit in the next slide when I have kind of a figure to show you. But that tells us also that alvin count was not necessarily the most accurate predictor of survival, because we had seen in the field when we were collecting them. And then also we had some lab observations that the alvin were actually escaping through the side of the tube. So they were swimming out through those small holes, even though theoretically they shouldn't have been able to fit. They're just really wiggly and able to kind of contort themselves through. So counting the number of alvin in the tubes was just overall not going to give us an accurate number. But counting the number of dead eggs, we found that throughout our monitoring visits, it was pretty accurate in evaluating the survival, because those eggs stayed throughout because the water was so cold. They weren't decomposing. They weren't going anywhere. So we could count the number of dead eggs and have that confidently know that that was a representative of the survival in the tubes. So again, kind of looking at that comparison, you can see these graphs are more or less opposite from each other. So the alvin count and the survival estimate, which is based on the egg count, are kind of flipped. So going forward, we only used the egg count to represent survival and run all of our other analyses and stuff because we were more confident in that number. And we ran some different models and analyses, and basically all that I'm going to summarize for you here is that of the different things that we looked at, so the temperature, the groundwater, the proportion of fine sediment, we found that only number of days freezing, so number of days below zero, had a significant effect on the survival of the alvin, and it was a negative correlation. So the more days below zero meant lower survival in each of the tubes. So that's kind of interesting and helpful going forward. And then also you can see here, this is just a cool figure to show us between site 2 and site 3 and the different depths. This is just temperature, but it's just each individual temperature point plotted on the graphs. But you can see, so site 2 had higher survival, and site 2 never really dips below zero. But then site 3, there was a couple freezing points, and they're very obvious, and depth doesn't really protect against that. So putting them deeper didn't really protect the eggs from the freezing necessarily. So it's interesting that kind of that freezing had such a big influence, but the other things didn't necessarily. That's not to say they wouldn't in other sites, but in this site specifically, we only really noticed an impact on survival by freezing. So then just to kind of sum up what we found and kind of what we're doing with this information. So we had overall just over 70% survival, so 71.4% survival in all of the tubes, which for context, we sent some eggs to the hatchery and they produced an 85% survival to post-emergent. So that kind of gives us a really good idea that 71% is a really good number for wild reared bull trout in stream in conditions that the environment throws at them. And it also tells us that site 2 had higher survival, and site 2 actually was also the location of higher historical red density. So from those red surveys from the 90s to the 2000s, site 2 had higher red concentrations. So it's interesting that maybe we can just follow in streams where we want to repopulate, follow where the bull trout have been, because it seems like they're pretty good at deciding where to put their eggs. And then also that freezing of a site is significant, and that, among other things, we should focus on going forward. So future site selection and deciding where we should put the eggs in the future could also potentially be done by monitoring the site for at least a year and seeing which sites freeze and the temperatures of those sites and the drying of the water bodies and the amount of ice cover. And that would kind of give, based on what we found, a good idea of the survival and success of these individuals going forward. And for example, some of our temperature loggers and even possibly some of the tubes in the creek, so we did the deployment and everything. The loggers we put out in spring, which obviously is the highest water levels, and then even the tubes we put out in early fall, so about late September. And it ended up being that after winter, some of those tubes were actually no longer underwater, and some of those temperature loggers were also no longer underwater or under deep sheets of ice and snowpack that we couldn't even get through. So, some kind of future planning and monitoring of these sites could help to avoid those kinds of hiccups that we ran into. And then we also, out of this, decided that trench technique is actually really good. And even though it was kind of an accidental kind of analysis, it for future use, putting them all together, it saved so much time. It had slightly lower survival, but that could have been caused by various factors, like they were sitting in the cooler for longer, and we didn't necessarily dig them deep enough the first time round. Overall, it was much quicker to get them in the creek. It took about 20 minutes for the whole trench, so 72 tubes, and for 36 tubes to be put in individually, it took about 3 to 4 hours. So it allows for more tubes in less time, and from a management kind of conservation, translocation, restocking type of perspective, that is kind of what we're looking for. We're looking for something more usable. And then there's a few other points, like making the holes slightly bigger so that maybe in the future we don't have to go out and time our retrieval exactly with when the alvin are hatched and ready to go in the stream. But we can just time retrieval to go collect the plastic at the end and let them swim out on their own so that we're not handling any fish and not impacting them. And then we can just count eggs for survival estimates because we know that that was a reliable option. And then in future, kind of some recommendations are trying places where bull trouts currently face some barriers like waterfalls, and kind of evaluate an area based on some pre-existing framework and decide, could bull trout actually survive here? Is this a good idea to move them here? And then there's options like assessing survival to maturity in fishless streams because there would be no other fish there to compare with. And then from a management perspective, it's just kind of a great option. And just kind of the significance of this. I think that it's a great option and strategy for future translocations going forward and for kind of improving bull trout populations in the province. There's the potential to apply it to kind of various streams and other populations, pending some kind of future research and checking off some boxes. It's not a universal solution, so we can't just use it to solve all of our problems, but it could be a good tool to add for helping this population. And I think that might be all. I think I might have some pictures, maybe not. Yeah, I think that's it. Thank you.
Thank you, Tara. I've got lots of questions, but are there any others in the audience that might want to ask a question? Then we'll flip over into another presentation. Dean.
Yeah. Thank you, Tara. There was a lot of information, and I'm sorry I missed parts of it. The difference between Site 2 and Site 3, can you summarize what they were and why you think Site 2 was so relatively successful?
Yeah, definitely. I know it's definitely a lot of information to kind of throw at you and to not be there to kind of elaborate. But so they were all within the same stretch of creek, and we were able to walk between them. But we didn't. We drove because we had those eggs and lots of equipment. But so Site 2, which had the higher survival, had, when we went and monitored them, more running water throughout the year. They didn't freeze as much. There wasn't as much snowpack. Versus Site 3, we thought it was really great when we went and saw it in the spring and summer. But then as kind of the year went on, there was a lot more freezing of the site and quite deep sheets of ice and a lot of snowpack above places where there was running water when we put the tubes in, and then it just froze over. So that was kind of the main difference between them, is that although site 2 might have been slightly similar and they were very close together, one just froze a lot more than the other.
And I missed a conclusion about depth. Was there a big difference in depth of where you placed in survival or outcomes?
So no, basically to sum it up, there wasn't. We had found previous research, somebody had done something before and said anything deeper than 10 centimeters should survive. They'll freeze if they're at 10 centimeters and they'll all die, but anything deeper should be okay. So that's why we kind of focused on the 20 and 30 centimeters, because we knew that 10 centimeters was a bad option. But based on what we found in the survival numbers, there was no difference between that 20 and 30 centimeter depth. So as long as you kind of hit that threshold, it was all similar survival.
Thank you.
Tara, you used the terms tube and capsule. Are those interchangeable, or were those actually different things?
No, those are the same thing, and I had that problem in writing my thesis and everything else, because some people refer to it as a tube and a capsule, and I have to keep flipping it in my brain. But they're the same thing. They all refer to that red hockey puck shaped thing.
Right. And the fertilization was all done on site. Did you use one of those remote incubation trailers, or was it simply just underneath the tent there that we saw?
It was underneath that tent. We had to kind of do some hiking and bushwhacking to get down to the stream. So we made a makeshift tent, had the setup, and brought the portable ultrasound down, and had hatchery staff with us there to do the fertilization. But it was all done kind of with the feet next to the stream.
So the fertilization, the eggs into the capsules into the water, that's all a one-day exercise?
Yeah, so we caught the fish the day before, and then everything else happened in the same day.
Right. Did you have your temperature loggers? Did you have one per capsule or one per set? How many temperature was the ratio of temperature to capsule?
Yeah, so the temperature loggers we put out in before we put out the capsules. So it was based on kind of our predetermined site mapping and not necessarily what we were doing that day. But we ended up kind of with that initial 6x6 that I showed you on the retrieval map. So there was 6 sites, or we called them clusters, within each site that had 6 tubes. So let's talk about site 2, for example. There was 6 spots in site 2 that also had 6 capsules in them. Each of those individual spots had one. So there was one set of temperature loggers per 6 capsules, which they were pretty close, within a meter of each other. And then there was one at surface, 20 centimeters and 30 centimeters.
Okay, good. Go ahead.
Yeah, hi Tara. My question is back to site number 2. Seeing that it does seem like freezing is really the key success criteria. Is there any, I'm sure you mentioned it, but there was no evidence then of groundwater input in site 2?
So other research has found that groundwater input is very important for bull trout survival, but we found no difference in the groundwater input between site 2 and site 3 and how it impacted survival. I'm sure it might be more relevant in places where there might be more freezing and less freezing, but the site 2 seemed to have pretty decent flow of water year round. So it might have been a bit steeper. So maybe it didn't necessarily rely more on groundwater flow as other places that bull trout live. But groundwater, in theory, based on the literature, is supposed to have quite an effect on their survival. We just didn't find it to be specifically in Smith-Dorian Creek.
Yeah, oh, great. And then, so it's just the channel characteristics of that reach of stream then? Is that the deal? Can you predict it? I guess is what I'm wondering.
Yeah, so the site 2 was quite unique compared to the rest of the creek that we were kind of working with, which might be why the bull trout all chose there, based on like the red surveys in the past and everything. But yeah, I'm sure that we could do some kind of surveys of not only the things that we focused on, but also maybe vegetation or depth across the creek or width of the creek. A lot of things I'm sure play into it, but I only had unfortunately 1 year to look into this stuff, so we had to pick and choose what we thought would be important, and it ended up only kind of being one of those things.
Ken?
Hey Tara, thanks. Were the water flows monitored the year prior and the year after? To see if there's any kind of fluctuation in water flows?
No, we did not look at water flows, which I'm sure it would be very interesting to look at those, maybe in future work. But for this stuff, we did not.
Excellent. That was a really interesting presentation, Tara. Thank you very much. We're going to have a follow-up from Kenton now. He's going to talk about some of the what's next.
Well, thanks for having me, everyone. Thank you, Tara, for that. That was a really good presentation on the tool, and I'm gonna build on a lot of what you said. So I really appreciate the setup. My name is Kenton Neufeld. I'm a fisheries biologist with provincial government for the Rocky Mountain area, although I work out of the Cochrane office physically. This is Caitlin. She's fisheries biologist for the Cochrane area, so for the Bow Basin. And she's here to answer all your questions about.
Sorry, Caitlin. Caitlin was also working with us. We were working with Caitlin and her team actually on the Arctic grayling project a couple years ago, where we went to several sites and collected Arctic grayling and samples, and that was all as part of a research project that Jess and Caitlin were a part of. Sorry, I missed out that earlier. So
Yeah,
back to Kenton.
excuse me. Yeah, so Tara kind of did a great job of explaining one of the tools that can be used for bull trout recovery stocking. I'm gonna back up and take a broader view to start, and then zoom into a specific watershed, a specific stream, Upper Fall Creek, where we're looking at maybe applying some of these techniques and doing some bull trout range expansion stocking there. So yeah, start with a bit of a step back. I'm sure many of you have seen these slides or these maps and know this context, but I'll just kind of go over it quickly. So essentially, we've seen big declines in bull trout historically in the province. So on the left here is our adult densities, green being high, red being low historically. On the right is what we currently think they are. So big shift from lots of fish, lots of bull trout to fewer fish. I don't think that's a surprise to anyone here. The cause of that is it varies from watershed to watershed, but there's a whole bunch of cumulative effects that add up to result in that. So the big ones across the province are habitat impacts, mainly like sedimentation. That can come from different land uses, roads, trails, industry, forestry, like agriculture, all those combining to sedimentation issues. Historically, overharvest was a big issue for bull trout. That caused a lot of population declines. Fragmentation of habitat, either through dams or road crossings like hanging culverts, and non-native species. So brook trout compete and hybridize with bull trout, so that's the main one, but there's also brown trout and other introduced species as well. So all these species have combined to create the situation we're in now, where provincially bull trout are listed as threatened. They're listed federally as well. And we're really working to try and recover the populations that we can. And because these threats vary from watershed to watershed, And because there's all these different threats that are combining, the solutions for each population are going to be different, right? It has to be kind of a population by population approach. And stalking, the tool we're kind of talking about tonight, is one of those tools that can be applied in certain circumstances. Like Tara said, it's definitely not, it's not going to solve all the problems. It's not going to solve most of the problems for bull trout. But there are specific instances where it could help, and it could help us recover those populations or create more resilience in them. So that's kind of what I'm going to talk about a bit today. And I'm going to introduce our study watershed, our focal watershed, the stream that I'm really interested in and excited about. So this is Fall Creek. It is a tributary to the Ram River, which is west of Rocky Mountain House. I'm sure you all know where the Ram River is and have fished it. So this flows into the south side of the Ram River, below the north and south Ram confluence. So it's fairly low in the watershed. It has a waterfall on it about 7.5 kilometers upstream from the mouth. And it's a large waterfall, so fish cannot get above the waterfall. Historically, it was barren of fish above that waterfall. What makes it really interesting and important is it is probably the most important bull trout spawning stream in the area. So the big fluvial migratory bull trout that you might catch in the North Saskatchewan River or the Lower Ram River, most of those fish are going to this small section of stream to spawn. The AC have done some studies, some telemetry studies to look at movement, and they tagged fish that were spawning here and watched them disperse, and they dispersed all the way into North Saskatchewan, even up the Clearwater River a little bit. So these fish, so it just kind of shows how important this one stream is for a much broader population. It's been studied fairly well by the ACA. They did that telemetry study. They've done red surveys there for, I think, over 20 years, almost 20 years. And we've done a bunch of population monitoring through electrofishing for about 5 years out of the last 10. So we know quite a bit about it. Threw up some photos because it's always helpful to kind of see what we're talking about. That's one of our fish and wildlife officers. This was actually a fairly old photo, probably before it was closed fishing. So Fall Creek is now closed to fishing. This is the kind of the typical bull trout you would see spawning there. So one of those big fluvial migrant fish. There's also cutthroat trout in Fall Creek. Those are coming from the Ram River, historically stocked in the Ram and self-sustaining there. So those are coming up there. There's also some whitefish and occasional brook trout, mountain sucker, long-nosed sucker, long-nosed dace. And then the picture on the top right just kind of shows you in general the type of habitat. It's pretty canyony below the falls, very confined. There's lots of little ledges. Yeah, it's a really quite a beautiful stream, and it is quite unique in that bull trout make up probably two-thirds to three-quarters of the fish that you find there. So most of them are those juvenile bull trout, the young year that have hatched. The juveniles spend a year or two in the stream before migrating out to the larger rivers. But it's just dense with all these juvenile bull trout. So it's a really cool spawning nursery stream that's important for the wider area. It's been on our radar for a little while. Like we've known that it's an important stream for a couple of decades. Like I said, it was close to angling because of that, to protect those spawning bull trout. We've also known about some habitat issues in the stream. So partners like nonprofit partners and OHV groups highlighted some of the issues with some of the trails in the area that were contributing sediment into the stream and causing issues. So that spurred some action and some recovery work that culminated in 2018. So we we initiated a fairly large scale, not fairly, a really large scale habitat reclamation project at Fall Creek. So, and this wasn't just the government, Freshwater Conservation Canada, I might refer them as to you, just out of habit. Trout Limited, ACA, the government, federal government, DFO, we all pitched in and worked together to reclaim this OHV trail that was crossing Fall Creek. It was about 11 kilometers of trail with 54 crossings. And a lot of those crossings were right through spawning habitat. So not only was sediment getting into the stream, but there were literally wild tracks right through reds. So a really poorly placed trail. We reclaimed the trail, stabilized the banks as well we could, just using kind of simple simple like what's the term? Bioengineering. Yes, simple bioengineering techniques. Planting willows, planting native grasses, things like that. We estimate that the sediment input into the stream reduced by about 12%. Which is pretty significant for, you know, a single project in a watershed. We thought that was a pretty good effect. This is just a kind of a photo of one of the crossings on a tributary to Fall Creek to show you what we were doing. On the left is the before, the right is the after. You can see you know, the steep approaches into the stream, the bare soil. Anytime it rains, it's just gonna puke sediment into the stream and it'll be good. But this is a year and a half later, the picture on the right. So it didn't take long with a little bit of work and blocking off access, and it's grown in quite well. So we, like I mentioned, we didn't do this alone. We partnered with a bunch of different folks. This is just a slide to highlight that. I do want to emphasize that was a really important part of this project. Like, we couldn't have done it by ourselves. Yeah, I mentioned those partners already. We did monitor the population as we were doing this restoration. So this is a bit of a busy slide, so I apologize for that. But it's showing just the trend in adult or subadult/adult bull trout and immature bull trout over 5 years from 2018 to 2022. Two kind of takeaways to take from this. So one is the general increasing trend. So the blue line is the trend. So over that time, we saw an increasing trend in both size classes of bull trout, which is great. The other takeaway is to look at the reference points. So those colored horizontal lines are our FSI categories. So kind of our references. What you can see on the left there is that the subadult and adult bull trout, even though they're improving, they're still in that orange category, which is our high risk category. So they're improving, but they're still at a low density compared to what we would want from like a healthy population. The immature bull trout are kind of an anomaly. They're at a very high density relative to other reference populations. And that is largely because this is the nursery for that broader population, right? So right now, all those immature bull trout are congregated in Fall Creek. They haven't yet dispersed to the wider Ram or Saskatchewan watershed. So it looks like, I mean, there are a lot of immature bull trout there, but that's not necessarily a sign that there's no risk to that population. Typically, we look at the adult densities as an indicator of whether the population is doing well or not.
It's the red line.
The red line, the one that kind of bebops up and down, that is the average each year or the median each year. So for instance, here, like this is the median count between all the sites we sampled each year. And then this blue line is just the trend based on that. So same here, you can see in 2022 that the mean actually decreased a little bit from the previous year. But when we do just the general trend over the whole time frame, it's the blue line. So I said this was a talk about stocking, and then I talked a lot about habitat reclamation. So maybe I'll explain why. So when we're looking at bull trout stocking opportunities, a key piece of that is making sure that wherever we're stocking bull trout is suitable for that species. So there's a lot of factors that can affect bull trout populations, like I mentioned. If those aren't addressed, there's really no point in stocking bull trout over top of those, right? Like if the habitat sucks, if there's a lot of sediment, if there's brook trout everywhere, stocking bull trout into that situation is not gonna fix the underlying issues. It's not gonna recover the population. And so I talked a lot about kind of the habitat work we've done, and I'm gonna talk a bit more about some more habitat work, because I think it's really important to understand that the stocking is one tool that we use, and it is effective in a context that's really important to understand. And that's the context of the habitat and the other conditions that are going in that watershed. So, but in this watershed, I think there is opportunity for bull trout stocking. So this is exciting. What we are considering now is, because the population is still in that high risk category, it hasn't gotten to the place where we want it. We're looking for other actions we can do in Fall Creek to help boost the population even further. And one of those ideas is stocking bull trout above the waterfall to create a self-sustaining population there. And that would serve a couple purposes. That would create overall like more resiliency in the Fall Creek population. So below the falls, there's only 7.5 kilometers. If something were to happen to that habitat, that would have big implications beyond just that Fall Creek area. If we can expand the population above the waterfall as well, there's a resiliency if some catastrophic event happens, or if brook trout invade lower Fall Creek, or something happens to that lower population, there's almost a backup upstream of the falls. So that it'll increase resiliency. It'll also have a bit of a trickle down effect where bull trout will move downstream. Some of them will move downstream of the falls and still contribute to that lower population. So this is a cool opportunity we're looking at. We're looking at it. It kind of is we think a decent idea for a few reasons. One of them is what Tara mentioned. So Smith-Dorian Creek it was a great study area for Tara's experiment, because it was so well studied, right? Like there was all those previous red surveys. It was accessible. We knew a lot about the population. Fall Creek similar in that we have a lot of background data a lot of it from the ACA surveys about where the reds are located, when they're spawning, how many fish are there. And knowing that about a source population for stocking, like a source where we can get eggs or fish to move, it really gives us a leg up and helps us get through a couple of hurdles. Because there's a lot of logistical hurdles and potential pitfalls when we're talking about stocking bull trout. One of the big ones is disease. So I'm sure you're all familiar with whirling disease. There's other ones out there as well. Whenever we're stocking fish or moving fish on the landscape, we want to make sure that we're not also moving diseases. We want to minimize that risk so that we're not doing more harm or any unintentional harm. So this year we actually went out to the Lower Ram River. We sampled a couple of tributaries, not Fall Creek, but a couple of other tributaries of the Lower Ram. And we sacrificed 60 brook trout, and we made some assumptions that those brook trout were suitable surrogates for bull trout, so that they had similar susceptibility to diseases as bull trout. We took those brook trout into the disease lab here in Edmonton, tested them, and made sure that there weren't any diseases in that watershed that we would be worried about transferring above the falls. And we got the results back. They were all positive, or they were all negative, I guess. No diseases of concern. So a clean bill of health there, which was great. So that's one of the hurdles that we had to go through here. And any project, when we're talking about stocking or transfers of fish, that's one of the considerations that we'll always have to think through. Another one is the genetic consideration. So this is a busy slide again, and I apologize for this. Essentially, this just shows the complexity of genetics that I don't even fully understand. We had our geneticist put this together. What we're looking for though, when we're thinking about genetics are the fish that we're moving hybridized? We don't want to put any fish that have brook trout genes above the falls. And is there enough genetic variation in that population so that the resulting population is sustainable? And are they locally adapted enough to fit that habitat that they're going in? So what this plot is showing is all these different populations in the red deer in North Saskatchewan and how closely they're related. Fall Creek is here, and you can kind of see the bigger the number, the more different it is from all these other populations. The smaller the number, the more closely related. And what this helps us do is pick out populations that are similar to Fall Creek that maybe we could also use as a source. So if we're establishing a population in Upper Fall Creek, we want a source population that is locally adapted to that area. And that usually means we're making some assumptions, but that usually means it's from a similar location, like close by. So, and that it's genetically similar to populations in that area. So that's what we're looking for here. I don't claim to be a geneticist by any means. And so we have one on staff, James Bull, who helps us walk us through these conversations.
Sorry to interrupt, I just wonder what's so special about the Upper Pine River?
Yeah, man, we could spend hours talking about this figure. That is really interesting. So that's Pinto Lake at the very top of the Cline River. And it's red because it's very different from all the other populations. There's a waterfall on the Cline River about 1.5 kilometers downstream from Pinto Lake. And that population has, it's been in Pinto Lake for a long time, and based on this anyway, genetically it's diverged quite a bit from the other populations. So yeah.
So they were stocked?
In Pinto? I don't think so, no. We don't have any records of stocking anyway. So they
Swam on the falls.
it's not a huge falls. It's about 2 meters high, and it's got a couple steps in it. So you can imagine maybe there's a flood event at some point, or an osprey grabbed a bird or a couple birds over the last couple millennia and moved them up. So yeah, we have no records of stocking. We actually have anecdotes of Indigenous people fishing for bull trout up there like centuries ago. So they've been there for a long time. It's a really cool population. It's close to fishing as well because it is so unique. But that's a whole other topic. I'd love to chat more about Pinto Lake with anyone after this. So as we're talking about moving bull trout into Upper Fall Creek, kind of getting back to that habitat suitability bit. So we've already reclaimed that one trail, which was mostly below the falls, but there's a bit above the falls as well. We are looking at doing more habitat improvements. FCC is actually gonna spend a considerable amount of effort in 2026, this coming year, to reclaim a bunch of trails in Upper Fall Creek. This is just one of those, picture of one of those trails, probably one of the worst ones, where there's been a lot of erosion. A lot of that sediment has gone down the hill into Fall Creek. So FCC is gonna be, I think, reclaiming 30 or 40 sites. And that just gets back to making sure that the habitat that we're putting bull trout in is the best possible, so we can give them the best chance of succeeding at the year. And then this is kind of scoping back in a little bit, back to some of what Tara was talking about, the like the how do we move the fish? There's a lot of unknowns with stocking bull trout. We don't do it very often. And we very rarely do it for conservation purposes, like, excuse me, in Alberta. I'm not sure if we've done bull trout stocking for conservation. We've moved a few populations around, but more just recreation or because we could. Even outside Alberta, it's quite uncommon to stock bull trout. And so there's a lot to learn, and that's why Tara's research was so important. But there's also other pathways of stocking that we could explore and test out. And we're hoping to try out a few of these in Upper Fall Creek in the next few years. So let's go through them here. So one of the first ones, this is what Tara was doing. So you catch the adult bull trout, collect the eggs, they go into these capsules, which are buried in the sediment of the stream, hatch into alevin, which then swim out into the recipient habitat, which in our case is Upper Fall Creek, and hopefully survive and do well there. One deviation from this pathway is after we collect and fertilize those eggs, we can actually bring them back to the fish culture facility. So in the Crow's Nest Pass, we have Allison Creek Brood Trout Station, which has some quarantine facilities. So sections of the hatchery that are completely isolated from the other portions. And that's important so that if we bring wild fish or wild eggs in, we're not risking contaminating the whole thing with disease or anything. But we can bring those eggs into that facility, hatch them in the facility, and then once they're hatched, stock them back out into Fall Creek. And there's pros and cons to each of these two. One of the pros to the capsule method that's buried in the stream is that it really it starts off those fish with the experience of incubating and hatching in the stream. So they have that experience of, the water and the sites are where we want them to stay and live. We want them to come back there to spawn themselves when they grow up. By hatching them in the hatchery, we lose some of that. So there's a bit of hatchery influence there that isn't ideal, but the trade-off is it's a controlled environment. We'll likely get much better survival, or we'll get good survival, consistent survival in the hatchery. We might get good survival in the wild if we use those capsules, but in the case of Fall Creek, unlike Smith Dorian Creek, we don't know where bull trout would normally spawn. We don't have in Upper Fall Creek anyway, we don't know where the bull trout would like to spawn. We don't have that fine scale information. So we can guess based on things like temperature and groundwater and all those other factors that were discussed earlier. But it's a bit more uncertain. So we might not get the same survival that Tara did for her study. So bringing some fish to the hatchery really is like it's a bit of a safeguard, not putting all your eggs in one basket type deal. The third way that we could do it is just by moving fish. It's the simplest. So we could go downstream of the falls, electrofish, collect a bunch of, these are just young and beaver bull trout, collect a bunch of bull trout, and essentially put them in buckets, carry them up the trail, up the falls, and put them in the stream upstream of the falls. So that's the simplest route. And I think if I get kind of what I hope to happen, we will actually try that as well. The benefits are that it's logistically way simpler than collecting the eggs from the fish. It requires so much less staff time and less resources. And we know those fish are already live fish. Like, we're not worried about the egg hatching survival. We know they're live fish. We know exactly how many we're going to put there. There are risks though. There's risks of when you're removing live fish, the risk of disease transfer is higher. So even though we tested the lower red population, there's still the risk that there could be some disease there that we missed. So if we're moving these fish from below the falls to upstream, we can't test them before we move them, but we're taking a bit of a risk that there could be something going with them. Same with the genetics. We could take fin clips from the fish that we move and test them later, but we won't be able to test them until after they're already released. So that's a bit of a risk, whereas when we're collecting the eggs, we can take fin clips from these adult fish, test the genetics, and we'll know exactly what genetics we're putting into Upper Fall Creek before we get to the stage where the eggs hatched and the elephants are in the stream. So a bit of trade-off, but this is because it is such a new experience for us and for, I mean, fish management in bull trout range. Like I say, not many people stock bull trout, and there's a lot to learn. I think doing kind of these 3 different methods would really help us figure out how to do this better, and that would have implications for doing it in other places too. So the more we can learn as we do this, the better, I think. Yeah.
Would you monitor the adults if you put them in?
So we wouldn't be moving adults. So we would collect the eggs from the adults. And then-
At number 3, the bottom.
Yeah, so these ones we would do monitoring for sure. Regardless of what technique we would use to stock the fish, we'd monitor Upper Fall Creek. So that would consist of probably electrofishing surveys to see what the abundance is of bull trout upstream of the falls. And we'll also know the genetics of for each of these different methods. And if we keep them separate, we can look at the genetics of the fish that we catch later on in Upper Fall Creek, and we can tell exactly which method they came from. So in 5 years, if we catch a bull trout in Upper Fall Creek and we take a fin clip and send it off for analysis, we'll be able to tell if that was one of the ones that we moved here, or if that was one of the ones that came from the hatchery or the egg capsules. And so that's really the way we would evaluate the success of this, to figure out which of those techniques is best.
You get an idea where they spawn too, and if you move adults.
If we move adults, would we get an idea where they spawn? You could do that, like, yeah, so move them into Upper Fall Creek and see where they go. It's interesting. The reason why we don't want to move adults is by moving adults, you're taking a fairly large chunk of the population, even just if you're moving a handful of adults. So we figure there's in the range of 50 to 100 adult fish in the Lower Fall Creek population, so not a ton of fish total. If we collect eggs or move the juvenile fish, that's a lot. There's a lot more eggs. There's a lot more juvenile fish in the population. So we can move, we're talking the order of like, maybe we move 1 to 200 of these fish out of thousands. If we look at the adult fish, there's only going to be, yeah, like maybe 100 fish in the population. So we could only move a couple without having a sizable effect on the population. So it's a bit of a balance act, trying not to impact the source too much. And that's why we would move the adults. Yeah.
Nice.
Yeah.
Have you got any estimate of the relative cost of the 3 methods?
Not in monetary terms. I've done a bit of analysis in terms of how many like staff days it would take to do each one. I'm gonna exclude the hatchery staff time, how long it takes to incubate the eggs, because I haven't really looked into that too much. But this could be done, you know, if you had a week and 8 people, you could, I think, quite comfortably move however many juvenile fish you need to from below the falls to above the falls. For these methods, it would be probably 3 or 4 times that much in total. Because you would have to go ahead of time to monitor the spawning run to make sure you have the timing down. You'd have to capture the fish. Tara did a great job explaining how much work it is to actually install the capsules in the stream. And then you'd want to go back and check on those throughout the incubation as well. So there's a lot of kind of pre-work and post-work that comes with the other methods. Yeah. But I haven't broken it down to dollar amount yet. I don't know if I want to. That's a little scary. Yeah, so I mean, this is next steps, we kind of talk about this. So the next thing, we haven't done this yet. This is kind of in the conceptual stage. I'm just working on putting together a implementation plan for this. So working out those details of like how many staff days it would take, when they would be like putting dates in calendars, putting names to those dates. Working with our partners, we have a project team that consists of ACA, Foothills Research Institute, Freshwater Conservation Canada, and I guess that's it for now. But we meet regularly to talk about this project. And so we're working together to put together a plan to actually to hopefully get it done, at least in some form, next year. Like I mentioned, Freshwater Conservation Canada is planning on doing a bunch of habitat reclamation work next year. So they've got the funding for that, they've got the plans for that. So that's hopefully going ahead. And then hopefully we'll be able to actually implement this at least the first year, starting next year, if all goes well. And I anticipate it'll be maybe like 3 years or so of stocking. So we wouldn't want to do it all in 1 year. That would just spread out the impacts to the source population a bit. And, you know, if there's some catastrophic event, it'll be spread over a few years, which will help things out too. So hopefully next year is the first year of 3.
So is forestry, put any, action into any of this stuff? Because there's a lot of, they're cutting up in that area.
Mm-hmm. Yeah, we haven't talked to them specifically about this project. They were, they have been very supportive in some other bull trout recovery work we've done in the area, like Southern Forest Products has been. So we haven't talked to them specifically about this area. I mean, we would get in using their access road. I think most of their harvesting is complete in that watershed. It has been harvested quite extensively. But I think most of their harvesting is complete. So there are conversations with forestry about bull trout recovery and native trout recovery and offsetting and all those things, but we haven't gotten to that specific level with this project yet. So yeah. Mm-hmm.
You mentioned that there's no fishing in Fall Creek anymore. Are the HOV trails still open? OAG trail still open?
Define open. We did our best, so it's not in a public land use zone. So there's no designated trails there. We did our best to physically make it extremely difficult to use those trails. So we had some legislative tools initially, so we could implement a short-term closure when we did the reclamation work, but that wasn't a permanent solution. So since then, we've been relying on those physical barriers. So we built some big berms at pinch points, felled logs, to physically prevent the quads from going in. We actually just had a chat with ACA this morning, and they've been in there this fall, and they said it was actually, seems to be working. There's not really any OHV use on the trail below the falls at this point. So in this case, it seems like it's been successful in keeping motorized traffic out of that former trail. But it's not closed. If that makes
And you're
sense.
gonna stop above the falls and that's still open?
Again, there's no designated trails there. There are potentially OHVers could get in and utilize some of that area. But Freshwater Conservation Canada is going to be kind of doing similar work upstream of the falls and trying to prevent or limit access on some of those former trails through the reclamation works. Yeah.
Thanks, Ken. I just was fascinated by that slide on the post remediation work. I just want to see if I understood it. So on the young fish, there was a fantastic response. You know, post remediation. I had that right? That there's really high densities of great, 1 and 2 year old fish.
This one?
Yeah,
Yeah, so the response is actually similar if you ignore the scales.
okay,
So subadult, adult bull trout and immature bull trout, they both increased over that time period, right? The difference is the absolute densities of immature fish are much higher relative to like the FSI scores, like our reference conditions. So yeah.
so it looks like it was always pretty good, but it went right off scale, really, right?
Yeah, again, though, this is where there's probably some issues with the scale that we're monitoring it at. So this Fall Creek is the rearing stream for a much bigger population. So we're only measuring kind of the one spawning stream in the whole population.
Mhm.
If normally when we assess populations, we would, randomly have sites throughout the whole, so AC actually did a bunch of monitoring in the rest of the lower Ram and all those other tributaries, so the same population. And they found very few juvenile bull trout, so
Very few,
yeah. So,
and so it wasn't just like, whether it was perfect in this year, it's just, yeah, okay.
no, so this is, to be honest, I probably, the figure's a bit deceptive without a lot of context in terms of the absolute.
That's okay, and I just wonder, like, so you think you're gonna continue to see recruitment into the subadult population then?
Yeah.
So that trend will keep going and, I mean, you already jumped one FSI score, so that's pretty fantastic, really.
Yeah. Yeah, like this is really what we usually base our population assessments on, is the adult fish, so. Yeah, this is the value you want to see it up to, moderate risk or low risk.
And so you believe you'll get there? Like in the trend, I just was wondering whether there was some, going out into North Saskatchewan and we do not survive, but that isn't what's happening, right? It looks like it's just slowly increasing and we'll get there.
I hope so. Like there is uncertainty for sure, because the stocking project is new. There's definitely no guarantees, but I think it has potential to improve the pop, and the habitat work upstream of the falls too it'll benefit the population upstream of the falls, but it'll also benefit the population downstream. So.
And so it's close to fishing, and you did this fantastic reclamation work that seems to really reduce the fees. Which, is there any way of teasing those two things apart?
The fishing closure was put in place quite a while ago. It was in early 2000s.
Okay,
Yeah. So the effect we're seeing here shouldn't be from that.
should be.
Yeah.
So, well, that's really a great model for other industries.
And this is, I know some of you might be familiar with the work we did in Rocky Creek as well, which is in the upper Clearwater. We did a similar OHV trail reclamation project there, and we saw a similar improvement in the population after that work. So these two systems have kind of demonstrated that reducing sediment, I should say, it's not about the OHVs necessarily, it's about the sediment that's coming in from those trails, right? So reducing the sediment into the streams, I think, can be shown to improve the bull trout populations.
And the adult bull trout population that's at spawning time or earlier in the year? Or is this skewed by the?
Yeah, so this is, we try to avoid the migratory periods when we're doing our assessments, just because there are those fish moving around. So this was, this data was collected in July and August, so before the migratory movements happened. Yeah.
I have one more quick question.
Sure.
Like, you mentioned some of the disease and genetics criteria for the translocation. Like, is there forage fish above the falls? Like, are they gonna be the bug eaters up there?
Yeah, I didn't, there's some details that I haven't gone into. I did mention this there. So we actually stocked cutthroat above the falls in the 80s or 90s. So
It will be cutthroat.
there are cutthroat above the falls. it's kind of a. In some ways, it's it's helpful because one of the considerations when we're stocking fish like this is we would want to limit our impacts on. Our ecological impacts. If there were no fish above the falls, there would actually be, we would have to think about things like impacts to amphibians and invertebrates in those fishless systems. But because we've already put cutthroat above there, that's kind of already happened. So we're not as worried about that if we put bull trout up there as well. And because the cutthroat have lived there for decades now, that tells us something about the habitat up there as well. We know they can overwinter. We know there's food for them. The spawning question is still, that's kind of the main question, like whether the spawning habitat is good enough up there. But because cutthroat have survived there for decades, we know that most of the habitat features are there. I think that was my last slide. Yeah. But I'm happy to take more questions if there's any.